Bismillahirrahmanirrahim. Acid-base balance or homeostasis of blood pH or regulation of blood pH by Dr. Ramawi Ibrahim Abdullah. This is introduction. Sources of acids and bases in body. By the end of this lecture, students are intended to know and make well understanding of these following items: introduction, first; second, sources of acids and bases in the body; 3, what is acid-base balance; 3, mechanisms regulating blood pH; 4, significance of maintaining acid-base balance; 5, acid-base imbalance and their conditions; 6, diagnostic tests. As an introduction, acid-base balance is a physiological and biochemical mechanism associated to body blood pH. What is pH? pH is a hydrogen ion concentration. In other words, pH is equal to minus logarithm hydrogen ion concentration. Different compartment of human body has specific pH. So different pH levels are noticed in each different compartment of the body. pH has role in enzyme activity. What blood pH is altered? Why blood pH is altered? The reasons for the changes happen to the body pH. Various acids or alkalis by metabolic activities alters body blood pH. So when we add acids or alkalis by metabolic activities, this results in changes for body blood pH. Types of acids and alkalis adding during metabolic life processes are hydrogen donors. Bases are hydrogen ion acceptors, or give up hydroxyl functional groups in solution. Introduction. Types of acids in the body are volatile acid, fixed acids, and organic acids. Mentioning volatile acid can leave solution and enter the atmosphere. Example, carbonic acid. Fixed acids are those acids that do not leave solution. For example, sulfuric and phosphoric acids. But when concerning organic acids, participants in or byproducts of aerobic metabolism. So, through the aerobic metabolism, participants in our body products may yield or may result in changes happening, decreasing or increasing body pH. Acids and bases can be strong or weak. Strong acid or base is one that dissociates completely in solution, just like hydrochloric acid, sodium hydroxide, and sulfuric acid. These examples of electrolytes are completely ionized in solution. Weak acid or base is one that dissociates partially in a solution, like carbonic acid and other lactate and other molecules. Substances of body: carbonic acid, H2CO3, phosphoric acid, H3PO4, sulfuric acid, H2SO4. Organic acids like lactate, acetoacetate, and pyruvate. Examples for alkaline substances of body are citrate and bicarbonates. Acid-base balance. The pH. Base balance is a homeostatic mechanism. So one of the mechanisms that regulate acid-base balance, one of the homeostatic mechanisms that maintain maintains body changes, maintain change in temperature, maintain change in blood volume, maintains change in body fluids, maintain change in minerals, body electrolytes. Acid base is considered one mechanism of body homeostasis. It carried out to regulate the altered pH of blood and other body compartments to its normal constant range. So this range where increased are going to be decreased and vice versa. Acid-base balance is a prime requisite to maintain normal, healthy, and active life. This balance is the regulation of hydrogen ions. The more hydrogen ions, the more acidic the solution, and the lower the pH. And vice versa, the less hydrogen ion concentrations, the more alkaline solution and the higher pH. The acidity or alkalinity of solution is measured as pH. Base balance regulates pH. So why it is very essential to regulate pH? What is the significance of pH regulation? pH blood and other body compartments are precisely regulated. pH is always tried to be maintained to its normal constant range. This maintains the blood pH at normal constant narrow range, minimum of 7.35, maximum to 7.45. The medium directly affects the enzyme activities. Optimum pH is an essential requisite for enzyme activities and normal metabolism. Prerequisite for regulating blood body pH, maintenance of normal optimal enzyme activities. Normal metabolism is also achieved by achieving normal body blood pH, also normal coordination. Besides, normal health is also achieved by maintenance of blood body pH within the norm. Factors regulating acid-base balance. Here you can see the lung. Here you can see the erythrocytes. Here are the kidneys. Balance is regulated by first line of defense, blood buffer system. Second line of defense, respiratory mechanism, and third line of defense, the renal mechanism. Chemical buffers react very rapidly, less than a second. Respiratory regulation reacts rapidly, seconds to minutes. Renal regulation reacts slowly, minutes to hours. So, considering the time factor, renal regulation need more time, or so as to be achieved takes minutes to hours. Considering respiratory regulation, to be achieved needs seconds to minutes, but buffering system is should be maintained within less than a second. Within less than a second, the buffer system should be maintained, or there may be detrimental health effects. Role of body buffer system is first line of defense, a mechanism of acid-base balance. Then acid's concentration of protons added are neutralized by the salt part of buffer. Bicarbonate buffer. This is sodium bicarbonate and carbonic acid is a buffer system constituted of a weak acid and its conjugated base. pH buffering range is at 7.4 pH. The phosphate buffer system is constituted of dihydrogen phosphate, sodium dihydrogen phosphate, and sodium dihydrogen phosphate, and the range of 7.4 pH. Protein buffer is sodium protein and hydrogen conjugated protein. Hydrogen protein and sodium protein is constituting the protein buffering system. Bicarbonate buffers, intracellular buffers, the buffers within the cell are bicarbonate buffers, phosphate buffers, protein buffers. Bicarbonate is constituted of carbonic acid and it is weak base, and phosphate buffer is constituted of potassium dihydrogen phosphate, and protein buffers and it is conjugate base, and protein and it is conjugate protein, hydrogen protein and its conjugate base. This acid and its conjugate base, the acid and it is conjugate base, the acid, weak acid and it is conjugate base. Action of buffer systems. Buffers mixtures are mixture of weak acids and its salts, or weak bases and their conjugated salts. Resist change in pH blood. So changes in blood pH are resisted by buffering systems when small amount of acids or alkalis are added to the medium, but when the large amounts of acids or bases are added to the system, there may be a collapse in maintenance of the system pH. Buffers act quickly but not permanently. Bicarbonate buffer system is a respiratory buffer system. Acid-base balance is primarily concerned with bicarbonate buffer mechanism. You can see this is hydrogen, proton, and this is a weak acid, carbonic acid, and this is the conjugate base of the carbonic acid, which is an alkali reserve. This is the conjugate base and this is the weak acid. Bicarbonate buffer, chief buffer system of blood. The main blood buffering system is bicarbonate buffer. Sodium bicarbonate is the salt part of buffer neutralizes strong and non-volatile acids added to blood. So non-volatile acids, when added to blood, the salt part of buffer neutralizes strong and non-volatile acids. So when non-volatile acids are added to blood, the bicarbonate buffering system is responsible for the maintenance of changes made to the pH by these volatile acids. It constitutes alkali reserve in the form of bicarbonate anions or bicarbonate functional group. This is a functional group which is named as bicarbonate. Carbonate buffer system enables the maintenance of body blood buffer in the norm through this mechanism. Here, carbon dioxide and water are combined, resulting in carbonic acid. Carbonic acid is reversibly dissociated into proton and bicarbonate ion. Here, the third step is bicarbonate reserve. Sodium, you can see sodium ions and sodium bicarbonate, reversibly bicarbonate here, going to go here in this phase, combined with the proton, results again in carbonic acid, again resulting in water and carbon dioxide. This phase, you can see carbonic acid bicarbonate buffer system. Here in this phase we see bicarbonate reserve. So bicarbonate here is dissociated into sodium ions and bicarbonate ions. This is cation and this is an functional group, is an anionic functional group. This is bicarbonate here and proton. Fixed acids or organic acids add protons. Protons are combined with bicarbonate, resulting in increased carbonic acid. Increased carbonic acid is dissociated into carbon dioxide and water. Carbonate buffer. When concerning bicarbonate buffer, we can observe sodium bicarbonate and carbonic acid maintains a 20 to 1 ratio, 20 for bicarbonate and 1 for carbonic acid. Here in this equation we can see HCl is combined with sodium bicarbonate reversibly. This is a reversible reaction yielding this molecule carbonic acid and sodium chloride. So sodium chloride and carbonic acid can result in sodium bicarbonate and hydrochloric acid, and vice versa. These two reactants can yield into these two products. We mentioned the left side equation, reactants, and the right side of the equation, of the chemical equation, products. So these two reactants yield reversibly these two products, sodium hydroxide and carbonic acid, reversibly resulting in sodium bicarbonate and water, and vice versa. These two products react to give reversibly these two reactants. Action of bicarbonate converts strong dissociable acid into weak non-dissociable acid. This carbonic acid is a weak non-dissociable acid, and a neutral salt without altering the pH. No change can be observed in body blood pH when bicarbonate converts strong dissociable acid into a weak non-dissociable acid. like carbonic acid formed during buffering action of bicarbonate, buffer is then expired out by lungs. Thus, bicarbonate buffer is connected to the respiratory system. So, bicarbonate buffer is also termed as respiratory buffer. Alkali reserve is represented by the concentration of sodium bicarbonate in blood. Alkali reserve concentration, the form of bicarbonate, determines the strength of buffering action towards added protons in proton ions by acids. So proton ions entered for the body system are reversibly maintained by the buffering system of carbonic bicarbonate buffering system. More the concentration of alkali reserve, more is the buffering action, and vice versa. Again, here we can see alkali reserve concentration determines the strength of buffering action. So, buffering action strength is assessed as an alkali reserve concentration. More the concentration of alkali reserve is the more the buffering action, and vice versa. Are effective as long as the acid load added is not very high and the alkali reserve bicarbonate is not exhausted. So the blood buffers are effective as long as the strength of blood buffering system is considered effective in accordance to these two factors. First, the acid load added is not very high. Second, the alkali reserve bicarbonate is not exhausted. So when there is supply of bicarbonate reserve alkali and no acid load is added, that means the blood buffering system is effectively performing this function. Phosphate buffer, urine buffer. Phosphate buffer is a measure of the urine buffer. Phosphate buffer is this acid and its conjugate base are in the ratio of 4:1 at 7.4 pH, which is considered slightly alkaline pH. So the minimum range of the pH is 0 and the maximum range is 14. The pH 7 is the neutral pH, which is the pH of pure water. More than 7 is considered more alkaline, less up to 14, and less than 7 is considered to be more acidic up to 0. If the proton is reacted with hydrogen phosphate -2 functional group, reversibly yielding phosphate dihydrogen functional group -1. Hydroxyl also is reacting with dihydrogen phosphate, reversibly yielding water and hydrogen triphosphate. This hydrogen monophosphate, and this is hydrogen diphosphate. When hydrogen ions added, they are neutralized, fixed by this compound, sodium dihydrogen phosphate, which is an alkaline phosphate, and converted to sodium phosphate monohydrogen, which is an acidic phosphate. These acid phosphates then excreted out through kidneys as acidic urine. Phosphate buffer is connected to excretory system. Phosphate buffer, also termed as urine buffer, enters this buffered by the acid phosphate, which converted to these molecules are the same. We are going to mention them in an assignment so as to distinguish which is monophosphate and which is diphosphate. Later on, inshallah, this molecule is considered to be an alkaline phosphate. Excreted in urine, making it alkaline urine. So, alkalinity of the urine is achieved through excretion of this molecule by the kidneys in the urine. Buffers includes hemoglobin, work in blood, that perform their function in the blood. The carboxyl group gives up pH, and the amino group accepts protons. So, increased protons means lower pH, and acceptance of protons means higher pH. The imidazole group of histidine present in hemoglobin structure has buffering activity, has buffering capacity. The buffering capacity of the imidazole group group of histidine, which is found in blood hemoglobin structure performing buffering capacity. Of respiratory mechanisms. The respiratory mechanisms, one of their functions is respiratory system plays second line of defense mechanism of acid-base balance through exhalation and inhalation, respiration of carbon dioxide and inhalation of oxygen. The role of respiration in acid-base balance is short-term regulatory process. Here, this is a question here. Question is, can be added into assignments. Are there any exhaust molecules other than carbon dioxide which are excreted by the lungs? Is the question. A role of respiration in acid-base balance is short-term regulatory process. Hydrogen, this carbonic acid formed from bicarbonate buffer is exhaled out through respiratory system. So this is an answer of our previous question. Besides carbon dioxide, you can see here carbonic acid formed from bicarbonate buffer is exhaled through the respiratory system. Is it exhaled as carbon dioxide or as carbonic acid? You keep in your mind and add to your assignment. Increased carbonic acid stimulates the respiratory center and medulla oblongata. This is part of the kidney. This is medulla oblongata. This in turn stimulates hyperventilation, which promptly removes carbonic acid from blood by expiration. So the removal of bicarbonic acid is achieved by the lungs. Exhalation of carbonic acid is as carbon dioxide by activity of enzyme carbonic anhydrase of lungs. So this is bicarbonate and this is proton. Reversibly, this is carbonic acid, reversibly carbon dioxide and water. So this molecule is considered to be exhaled by the lungs out of the body, besides water. The mechanism is powerful, but only works with volatile acids. Volatile substances are substances that can be as gas molecules within low temperatures. Does not affect fixed acids like lactic acid. Lactic acid as fixed acid cannot be affected by the respiratory mechanism, which can be adjusted through respiratory mechanism. So one of the important mechanisms of maintenance of blood pH is mechanism through respiration. By changing rate and depth of breathing, the rate of the depth of bracing. These two factors, the rate and the depth of bracing, are going to manifest or adjust the maintenance of blood pH through respiratory mechanism. Low carbonic acid concentration in blood depresses the respiratory center, causing hyperventilation. In other words, slow and shallow respiration. This retains carbonic acid in blood. Center respiratory system fails, acid-base balance fails. So the failure of the nervous center respiratory system is followed by the failure of the acid-base balance. Generation of bicarbonate by red blood cells. This is an RBC. This is the plasma. Carbon dioxide entering an RBC, conjugating water using carbonic acid anhydrase, resulting in carbonic acid. And this is an enzyme for activating the conjugation of water and carbon dioxide within the erythrocyte, yielding or resulting in carbonic acid. Carbonic acid is going to be on the this is on the third step, dissociating into proton and carbonate and bicarbonate. Bicarbonate is going to be thrown out of the RBC. Chlorine ions are going to be entered into the RBC. The proton is going to be conjugated with hemoglobin, resulting in H hemoglobin. So the name of this enzyme is going to be added for a question as an assignment. So the lack of aerobic activity diffusion of carbon dioxide is proton buffered by protonated hemoglobin. Events in lungs and tissue. This is a lung. This is cellular lung. This is tissue. I'm considering this system as lung and this system as tissue. Initially, through the metabolism, carbon dioxide is produced. Combined with water, carbonic acid is formed. Carbonic acid is going to be dissociated into a proton and bicarbonate. The proton, this is an oxygenated hemoglobin, is going to be resulted in protonated hemoglobin. When a proton is to be entered here, and an oxygen is to be taken out from this oxygenated hemoglobin. The protonated hemoglobin is going to be transported to the lung. Protonated hemoglobin results in oxyhemoglobin or oxygenated hemoglobin. Oxygenated hemoglobin reversibly here. Oxygen is added and proton is going to be repelled out. The proton is going to be combined with the bicarbonate, resulting in carbonic acid. Carbonic acid is going to be dissociated into water and carbon dioxide. Subsequently, it is going to be excreted out by the expired air. This is the isohydric transport of carbon dioxide mechanism.